Welcome everyone. In the previous lecture, we discussed about generation of gas, biogas through microorganisms. In this lecture also, we will continue with the steps of biogas production and the advantages of biogas, right? So the name itself tells biogas means the gas is generated with the help of microorganisms. That is why we are studying under the chapter Microbes in Human Welfare. See, I have drawn the diagram of a typical biogas plant. Okay, first let us see what is biogas, then we'll get into the production of the biogas. Now biogas, it is a combustible gas, means when you burn it, you get energy. So that's why we call it as a combustible gas produced by microorganisms. That is why it is called biogas. Microorganisms, when do they produce the biogas? During anaerobic process or aerobic process, means in the presence of oxygen or in the absence of oxygen. They generate gas when they do anaerobic respiration. So biogas, shall we write down? Biogas is a combustible gas, means when you burn it, you get energy produced through microorganisms, right? So it is produced through which breakdown? Aerobic breakdown or anaerobic breakdown? Anaerobic breakdown. So I am telling, like, anaerobic means in absence of oxygen. Microorganisms anaerobically they break the waste biomass into biogas. Anaerobic breakdown of organic waste biomass. So we generate biogas, which is burnable gas, combustible gas, through anaerobic breakdown. Microorganisms will do it. So they'll break the organic waste biomass into gas. So it's good, right? So we are converting the waste into a gas, so which can be reused. Understood? Now this is a typical biogas plant. A typical biogas plant in NCERT, they have given how deep is the digester. This is the main vessel, which is called as digester. The digester is 10 to 15 feet deep, they will tell. In NCERT, they have given a typical biogas plant. A typical biogas plant has digester. Amma, what do you mean by digester? This big vessel, I wrote here 3. This is 3. 3 is digester. A typical biogas plant has a digester. In NCERT, what's the dimension of the digester? 10 to 15 feet deep. It is 10 to 15 feet deep. And it is a concrete cylindrical tank, right? So it is concrete cylindrical tank. It is made up of a concrete material, cement material. It is a cylindrical tank. Come on, tell me, what is biogas plant? Biogas plant, a typical biogas plant has a digester. What is the depth of the digester? 10 to 15 feet deep. And it is an airtight cylindrical digester. Now it is this. Now this is connected to 1. I wrote the numbers. Let us see that and then we will write down. This is 1 I mentioned. Here is 2. This is 3. This is 4. And here is 5. And this is 6. Now if we have to generate biogas, we need to mix the biomass. Organic bio waste we have to mix. Now this is the place where the cow dung or molasses or aquatic weeds, all those things, anything it can be any dried material. Okay, so usually we use cow dung in villages because cow dung is applicable, and that's why biogas is also called gobar gas. So take cow dung, or you can take any aquatic weed, or you can take any organic matter, or you can take the farming waste also, farming refuse also, in which organic matter is there, the straw and all. So take that and nicely mix it with water. So that is why this tank 1 is called as mixing tank, where we take the raw material and mix it with water and prepare a fine slurry, a fine paste. Mixing thoroughly to make a fine paste is called as a fine slurry. So when you are mixing here, when you are mixing the contents here, it should not come down. So you have a valve here. That valve will, if it is locked, then the contents will not come down. You lock it, and then the cow dung and the water is nicely mixed here to get a slurry. What do we get? we'll get a slurry here. Now after you make the slurry, that slurry is sent down into this one, which is called inlet chamber. The second one I mentioned here, it is inlet chamber. So when you open this wall, the slurry comes into the inlet chamber. Via inlet chamber, it enters into an airtight digester. This is a digester. Via this inlet chamber, the slurry, the cow dung with water paste, which is nicely mixed up, enters into an airtight chamber, which is called as digester, which is 10 to 15 feet deep, we wrote, right? Enters into the digester. Now in this digester, if it has to enter into digester, there's one more inlet valve here. This is inlet valve and this is outlet valve. Only when you open the inlet valve, only when you open this inlet valve, then only the slurry comes into the digester. After the slurry comes into the digester, you close it. You need to ensure that the inlet wall is closed and the outlet wall is also closed. Then only anaerobic conditions are maintained. Then only microorganisms can convert the organic matter into biogas, because gas always comes with fermentation. Fermentation is an anaerobic process. So that is why usually they'll keep it as airtight chamber, and as soon as they enter the slurry, they will close it. Outlet also will be closed. Now what happens inside when all this slurry gets collected here? When the slurry gets collected here, now you should not overload the tank also. Why we should not overload the tank? Because here gas gets generated and that gas should get collected on the upper space, right? So that is why 2/3 we will use it for dumping the slurry, and the remaining we will leave it for collection of the biogas. Understood? So now this is a place where biogas production will be done. That we will see the steps. After the biogas production is done here, after the biogas production is done here, now how do you know that the gas got generated? Microorganisms acted on this organic mass and they generate the gas. How will you know that the gas got generated? Means there is a gas holder. 6, I label it as gas holder. In NCERT also they have given this. In this gas holder, the gas gets collected. As the gas gets collected in this one and this one, it keeps rising up. It keeps rising up, it keeps hitting. So we'll come to know, okay, gas got generated, and this gas holder is connected to pipelines. The gas holder is connected to, you can see the pipelines. When you open this wall, the gas leaks out, the gas comes out. You can use this gas for cooking purposes, for lighting purposes, for electricity generation purposes. You can use it in villages, in the farms, for pump sheds we can use, for running engines we can use, all those purposes, right? So as the production is completed here, gas is generated, that gas raises to this layer, to this level. From here, it gets collected into the gas holder. This gas holder, when it takes the gas, it lifts up. When it lifts up, so it makes sound, and then we can notice that, and we can open here. We can open here and then the gas comes out. That is what is biogas. Understood, children? Now, after the biogas is collected out, what happens to the waste material which is present here? After the biogas is collected out, what happens to the waste material which is present here? This waste material is taken into 4. This is outlet chamber. 4 is outlet chamber. Open the outlet wall. Shall I write down here? This is called as outlet wall. And here we will write it as inlet wall. Right? When you open the outlet wall, then all the thick, hard material will be passing through the outlet wall into the 5th one. The 5th one is called overflow tank. From here, it comes into the overflow tank. In this overflow tank, we will dry it. In the overflow tank, we will dry it nicely. When it gets dried, now it can be acted as a very good manure for the agricultural crops. It acts as a manure. You can use it for the cultivation of the plant. See, this itself, what you have used is waste. From this waste, we have generated biogas. Even after generating biogas also, again, you are getting manure. How good it is, isn't it? So, it is used as a manure for the growth of the plants and agriculture. Amma, this is a biogas production. Let us see once again. So, 1, what is it? It is mixing tank. What do we do here? We take the organic matter and we mix it up with the water and we prepare a fine slurry. When you are preparing it, this should be closed so that it's not coming down. After ensuring that a nice thin slurry is prepared, then open this and send the slurry into the inlet chamber. 2 is called as inlet chamber. Now when the slurry comes to the inlet chamber, open the inlet valve and collect the slurry into the digester. What is digester? It is an airtight cylindrical tank. Okay, so in airtight cylindrical tank we pumped all that slurry and ensure anaerobic conditions by closing the inlet valve and the outlet valve. Now here what happens is exactly the 3 steps of biogas production will happen, that we will see it on the board. So one is called solubilization, second one is called acidogenesis, third one is called methanogenesis. So microorganisms help in solubilization, they help in acidogenesis, they help in methanogenesis, and they generate methane, which is a major component of the biogas. That gas which gets generated gets collected in the upper space, and from there it goes to this gas holder. See, in the gas holder they have given, it's collecting the gases, like major gas is methane, then next it has carbon dioxide also, it has hydrogen also, traces of nitrogen, oxygen also it has. All that gas gets collected here, and when it is collecting the gas, it uplifts, it moves up and down. When it moves up and down, so we can notice it, that gas is generated, it is there in the gas holder. This gas holder is connected to the pipes. Now this pipe, when you open, the gas comes out. That gas is used for various purposes, and all the waste material which is left out here, open the outlet valve, take it to the outlet chamber. 4 is outlet chamber. Through the outlet chamber, collect it in the outflow chamber. This is overflow chamber. Collect it in the overflow chamber. In the overflow chamber, nicely dry it, and you can use it as a very good manure. This acts as the waste material after generating the biogas, which contains lignin, which contains partially organic materials and all, will act as a manure which can help in the further growth of the plants. Understood, children? So we understood the process. We have discussed about the diagrammatic representation also. Now if it is a board question, if it is for your board exams, a typical biogas plant is a 3-marker diagram for your boards. You just need to draw and label it, you will get 3 marks. Okay, so this is a typical biogas plant, so where I have little enlarged to make the process more clear for you. In NCERT they did not give these walls, they did not give the inlet chamber, they did not give the outflow chamber. This they did not give us. They gave only 1, 2 and 3. This and this were not mentioned. So you can draw the way which is there in NCERT also. We can take this picture also. Now let us get into the process of biogas production. Now we were talking about the digester, the dimensions of the digester, and we are telling it is a airtight chamber. Now let us talk, what is the raw material? What is the raw material? Come on, tell me, what is the raw material? All the waste material, like we can take cow dung, which is the major one, which is easily available in villages. We can take cow dung. We can take farm refuse also. Whatever the farm waste is there, that we can take. And we can take the aquatic weeds which grow vigorously, right? Water hyacinth and all, water lettuce and all. We can take all the water weeds. Water weeds are nicely used here. We can take water weeds, all these things, right? So we can take cow dung, we can take farm refuse, we can take water weeds, which acts as a raw material here. Understood? Now to this raw material, the raw material, it is mixed with, come on, tell me. We wrote here, the raw material is mixed with water. So when you mix the raw material with water, we get a fine, thick slurry. We get a fine thick slurry. This fine thick slurry is now sent into the digester. Is now sent into digester. It is sent into digester via inlet chamber, right? Via inlet chamber we are sending into digester. Now what happens in the digester is the biogas production, which happens in 3 steps. Take a screenshot. We'll talk about the 3 steps also. Right? Understood, children? Now let us talk about the 3 steps process of biogas generation, which happens in the digester, right? Is it easy? Now we are telling that inside this digester, what is this? Digester. Now in the digester, in the digester. Biogas production. Biogas production occurs at 3 steps. It occurs in 3 steps. What is the first step I told? It's called as decomposition or solubilization. Let us see step by step. First step is called decomposition, or you can also call it as solubilization. Now, what happens in the first step of biogas production means facultative anaerobic bacteria. Facultative means they can survive in the presence of oxygen, they can survive in absence of oxygen also. So when we are closing it, initially little oxygen will be there. In that oxygen also those bacteria should survive. So that's why the first step is done by facultative anaerobic bacteria. Facultative anaerobic bacteria, examples if we want, we can take Pseudomonas and we can take Clostridium as examples. These Pseudomonas and Clostridium, what do they do? So what organic matter is present in this raw material means cellulose will be there, hemicellulose will be there, lignin will be there, lipid will be there, protein will be there. They secrete enzymes to digest the raw materials. They break the complex organic raw materials into simpler forms. That's what is called decomposition. They'll convert the polymers into monomers. That is what is solubilization. Shall we write down in decomposition and solubilization? What are the microorganisms? I told they are facultative. And I told they are anaerobic microbes. Facultative anaerobic microbes. Like, now if you want to see the examples for this category, Pseudomonas we can take and Clostridium we can take. Like, let us write down Pseudomonas, which is one bacteria, and other one is Clostridium. Now these bacteria, they secrete enzymes. Like, what are the enzymes these facultative anaerobic bacteria will secrete? They secrete enzymes like, we can tell lipases. Okay, so we can call cellulases also, and proteases also, right, and pectinases also. So what do the bacteria do? The bacteria secrete enzymes like lipases, cellulases, proteases and pectinases. Now they break the organic material. Lipid is broken down into fatty acids and glycerol. Cellulases will break the cellulose into maltose. Proteases break proteins into amino acids, right? And pectinases break pectin into simple sugars. They break them into simple sugars. Are you seeing? So these bacteria are releasing enzymes. These enzymes are converting the complex organic materials. What are all these called? They are called complex organic materials which are present in the raw material. The bacteria are secreting the enzymes to break the complex organic materials into monomers, into their respective monomers. All these are examples of monomers. And this is what is the first step called decomposition or solubilization, which happens here. So even when we take the food, what we take is starch, what we take is protein. So our digestive enzymes act and they convert them into simple sugars, right? In the same manner, whenever you are giving a raw material here, the bacteria which are present in the cow dung itself, we don't add any inoculum. The bacteria which are present in the cow dung itself, they secrete enzymes to break the complex forms into simpler forms. This is the first step. Take a screenshot. We'll go to the second step. Right. Now let's go to the second step. The second step, as I told, it is called acidogenesis. What is the second step called? Step 2 is acidogenesis. Now, who will carry out acidogenesis? Fermentative microorganisms. We can tell fermentative microbes. fermentative microbes. If we have to list down the examples of fermentative microbes, Propionibacterium, Acitovibrio are the examples. Fermentative microbes like Propionibacterium and Acitovibrio. These are some bacteria. What do? They convert monomers. They are converting monomers into organic acids. Since they are making acids, the step is called acidogenesis. So earlier once they converted complex food materials into simpler forms, monomers. Then these things, acidogenic bacteria, they're converting the monomers into organic acids. They convert them into organic acids, and the examples are Propionibacterium and Acitovibrio. Now let's go to the third step, the last step, which is called as methanogenesis. Now this is the exact process. Methanogenesis is where methanogenic bacteria are involved. Archaebacteria we call them, right? Methanobacterium we call them, right? So Methanobacterium, Archaebacterium are involved in methanogenesis. What do? They take the organic acids, and these organic acids are anaerobically converted into methane. So what do? So Archaebacterium converts organic acids. They convert the organic acids into methane, which is the biogas methane with some other products like carbon dioxide. We wrote here carbon dioxide, H2, H2S, nitrogen, oxygen. All these are the byproducts which get generated. Methanogenic bacteria, so which are present in the rumen of the cattle, which is present in the cow dung also. Anaerobically, they break the organic acids into methane. If we want to see some equations also, how methane is generated, we can write on carbon monoxide when it combines with water. Then CO becomes CO2 and it gets generated as H2. Now this carbon dioxide, it combines with H2. Carbon dioxide combines with H2, then it makes methane and it makes water. It makes methane and it makes, two water molecules. So in the manner, carbon monoxide is converted to methane or carbon dioxide gets converted to methane. We can take formic acid also. OH, okay. Formic acid, what is this, ma? This is formic acid. Formic acid also gets converted to methane and carbon dioxide. Formic acid is also converted into methane, carbon dioxide. We can write on water also. In the same manner, acetic acid also we can write, right? CH3COOH. What is this called? This is called acetic acid. Even acetic acid is also converted into methane and water molecule. Even acetic acid is also converted into methane and carbon dioxide, CO2. These are the steps which happen in methanogenesis. Now come on, let us see what are the three steps. Solubilization, acidogenesis, methanogenesis. In solubilization, facultative bacteria, they break the complex materials, complex organic materials into monomers. Followed by acidogenesis, fermentative microorganisms, they convert the monomers into organic acids. Third one is methanogenesis, where these organic acids are converted into methane. Like see, carbon monoxide got converted to carbon dioxide. Carbon dioxide got converted to methane. Formic acid is also generating methane. Acetic acid is also generating methane. So these are the steps of biogas production. This is biogas. Take a screenshot, then we'll talk about the significance of biogas. Right? So hope you understood the process. Now let us see the significance of biogas. Now from where are we generating biogas? We are generating biogas from cow dung. So earlier in villages, when they don't know this technology, they used to make cakes from the cow dungs and they used to burn it. And when you are burning it, how much air is getting polluted, how much smokes get generated, right? So it is not good. Or they used to cut down the trees, they'll get wooden blocks, and that with that wood they used to generate fire. That is also not good. So when we come to know about biogas technology, instead of using that cow dung cakes, they can use that cow dung here and they can generate gas which burns fast. It's inexpensive also for them because it is coming from waste material. Now when we have to talk about the advantages of biogas, let us see the advantage of biogas. The first one is no smoke. They are free from smoke and no foul smell. When they burn the cow dung, when they burn the wooden logs and all, some smell will come. But when you generate gas from that we don't get any smell. So no smoke also, no foul smell also. And in that cow dung, pathogenic bacteria can be there. Those pathogenic bacteria are killed off if you leave it like that. So that pathogenic bacteria will be there only. But here in this decomposition, what happening is the pathogenic bacteria are killed off. That's the second advantage. The pathogens present in, where are they present? The pathogens present in fecal matter, present in the fecal matter, are killed. So we are free from that diseases. And the third one is it is cleaner. Biogas is cleaner than using cow dung and wood. Biogas is cleaner gas than using cow dung cakes, and wood. And where do we use biogas? NCERT says that biogas is used for cooking, it is used for lighting, it is used for cooking purposes, it is used for lighting purposes, it is used for moving engines, it's used for running engines, we can write, and it is used for pump sets, right? For the pumps we can use it,